So hello guys, I'm welcoming you all on this YouTube channel. So in this video lecture we will going to continue the topic that I've already discussed with you, that is the polymerase chain reaction, and I have already uploaded part 1 of this topic, and if you have not watched till, so do watch and understand the topic. So in this video lecture we will study in detail the stages of PCR, right? What are the stages involved in PCR? I have already given you the raw or rough idea or the superficial points of these stages, and in this video lecture we will going to discuss the details, right? What are the things involved in every stage, right? And what are the processes that are taken place in each and every step of this process of polymerase chain reaction, right? So the first stage is the denaturing stage, and during this stage we generally take the template DNA and mix with all the core ingredients, and that mixture, that cocktail is heated to 94 to 95 degrees Celsius. So you can note here the temperature is very high, because if in order to amplify the template DNA, so we have to make that DNA single stranded. For that we have to raise the temperature, and that temperature will cause the breakage of hydrogen bonds, right? That hydrogen bond which is present between the bases in the two strands of the template DNA. So we want to separate that those two strands or that double stranded DNA. So we have to raise the temperature, and that temperature will break the hydrogen bonds, right? So in this case this step usually takes between the 15 to 30 seconds, right? Now second stage is the annealing stage. So what happens in this stage, the reaction is cooled, that reaction mixture is cooled to 50 to 65 degrees Celsius, and this pretty much low temperature, or we can compare with the first stage in which we have raised the temperature up to a maximum maximum range, right? But in this case this temperature is very much helpful. In what sense? This at this temperature, primers are going to attach to the specific location on the single stranded DNA by way of hydrogen bonding, right? So primers are nothing but these are the single strands of DNA or RNA sequence that are around 20 to 30 bases in length, right? So these are not long strands of DNA, these are short stretches, short strands of DNA, right? So these primers are designed in such a way that these primers are complementary in sequence to the short section of DNA on each of the sequence to be copied. So what happens? The primers are complementary, and in by which they can easily bind to the specific sequence of that DNA which you want to amplify simply, right? So primers serves as a starting or initiation point for the DNA synthesis, and the polymerase enzyme can only add DNA bases to a double strand of DNA once only the primer has bound, right? Primer once bound to that strand of DNA, the polymerase enzyme will add DNA bases, right? So the two separated strands of DNA are complementary and run in the opposite directions, right? So I will show you this stage or this process in coming slides, and I will show you the diagram of this particular reaction, this particular procedure. So this step usually takes about 10 to 30 seconds only, right? So these are all about the two stages. The first is the denaturing stage, and the second is the annealing stage. The last stage is the extending stage, right? So this is the final step, and in this case the heat is increased, right, to 72 degrees Celsius, and this temperature enables the new DNA to be made by using a special DNA polymerase, that is stack polymerase, and this enzyme is very much efficiently work at this higher temperature, and this DNA polymerase enzyme, Taq polymerase enzyme, is obtained from the heat loving bacteria Thermus aquaticus, because this bacteria normally lives in the hot springs, right, and it can tolerate even above 80 degree Celsius temperature, right. So the bacterial DNA polymerase is very stable at higher temperature, which means it can withstand the temperature needed to break the strands of DNA apart in the denaturing stage of PCR, right. After that, this polymerase attaches to the primer, and then it is going to add DNA bases to that single strand one by one in the 5' to 3' direction. And as a result, there is a brand new strands of DNA and a double stranded molecule of DNA is formed, right. And the duration of this step depends on the length of the DNA sequence is being amplified. मतलब कितनी length है, how much the length of the DNA that is going to be amplified, and but it usually takes around 1 minute to copy 1 kb base pair of DNA or 1 kb bases of DNA, 1000 bases of DNA को ये कॉपी करने के लिए it takes only 1 minute, right. So this is the diagram, right. You can see over here, this is the first stage. This is a mixture in which you have add nucleotide bases, right, or different enzymes, buffer, template DNA, right. So this is the denaturation stage in which the two strands are far apart, then primers attach, a leading stage, and after that the synthesis takes place, elongation stage, right. So you got two strands from two new DNA double strands from this one, and from these you again got two. So in this case there is a cycling, right. In this case there is a cycling occur. So therefore these three processes of thermal cycling are repeated 20 to 40 times to produce an ample of copies of the DNA sequence of interest, right. It's a kind of thermal cycling because we in every stage, in every step, we change the temperature, high, low, high. So this is a kind of thermal cycling, right. And after 20 to 40 times we go for this cycling or this pollination reaction, we'll got ample of copies of DNA molecule, right, and that DNA which is of our interest, right. So the new fragments of DNA that are made during PCR also serve as template. You can see the newly formed DNA also acts as template, and from that a new more strands of DNA are formed. So as a result, you got a huge number of copies of that specific DNA or the specific sequence which is of your interest in a relatively short period of time, right. So this technique is very very important and it is widely used, right. So with this your PCR reaction is complete. I hope you guys enjoyed the lecture, and also it definitely helps you in your exam, and wish you all the very best. Thank you.